Hi, I'm going to do a brief review of human genetics and then explain how to complete a simple Punnett square. Punnett squares are used by biologists to predict the possible outcomes for offspring of a breeding experiment. In order to complete a Punnett square, you must know the genotypes of the two parents involved. The genotype refers to the alleles that an individual possesses for a particular gene. Genes are located on chromosomes, which are shown here, and there are always at least two alleles that make up a gene. The interaction between these alleles will determine the individual's phenotype, which is the characteristics that they display as a result of the alleles that they have. Normally, alleles are either dominant or recessive. If an individual has two alleles that are the same, they are said to be homozygous, and the individual will always display that particular characteristic that these alleles code for. Here are two examples of a homozygous genotype. If an individual has two different alleles, one dominant and one recessive, then they are said to be heterozygous, as in this case. If this is the case, then only the characteristic determined by the dominant allele will be displayed. Generally, alleles are abbreviated using the first letter for the characteristic that they determine. For instance, if a particular allele determines the eye color brown, then it would be abbreviated with the letter B. Dominant alleles are represented with a capital letter, and recessive alleles are represented with a lowercase letter. If brown, for instance, as an eye color, were the dominant allele, then it would be represented with a capital B. The recessive trait, let's say for blue eyes, would be represented with a lowercase b. In this Punnett square example, we will look at plants with flowers that can either be red or white. Red is the dominant allele and will therefore be represented with a capital letter R. White is the recessive allele and will therefore be represented with a lowercase r. For this Punnett square problem, we have a plant that is homozygous recessive. This means that it has two recessive alleles represented with lowercase r's as its genotype. This also means that its phenotype will be white flowers. The other parent plant, number 2, is heterozygous. This means that it has one dominant and one recessive allele. Therefore, the phenotype for this plant will be red flowers. The two alleles for the first parent plant will be written above the Punnett square, and the two alleles for the other parent plant will be written beside the Punnett square. The Punnett square consists of 4 boxes. Then each box will be filled in by transferring the letters above and next to each box into that box. So this lowercase r has been brought down into this box and this box. This lowercase r has been brought down into this box and this box. Then the capital R was brought over into this box and this box. And the lowercase r was also brought over into the 2 boxes that it is next to. Now that the Punnett square is complete, you can see the possible genotypes for the offspring of these 2 plants. 50% of the offspring will have the genotype heterozygous. So they will have one allele for red flowers and one allele for white flowers. The other 50% of the offspring will be homozygous recessive. They will display 2 alleles, both code for white flowers. Therefore, the phenotype for this genotype will be white flowers. The phenotype for the 50% of the offspring who are heterozygous will be red flowers, because the red allele is dominant over the white allele.